So now irreversible cell injury. In irreversible cell injury, the mitochondrial dysfunction is important. The mitochondrial dysfunction, as we saw that the mitochondria is the first organelle which is going to affect it. And the another thing which is new to us is the necrosis. Irreversible cell injury, the cell is going to die. The cell is going to die. Okay, and the way it is going to be killed is by necrosis. Okay, now let's see first the mitochondrial dysfunction. What is, how is the mitochondrial dysfunction is occurring? So the thing which we need to be remembered that we know, I think we know this stuff. In the cell, inside the potassium is more, inside the sodium and calcium is less as compared to outside. Sodium and calcium is more outside. Okay, sodium and calcium is more outside. Okay, so when we talked about in reversible cell injury, the thing which we talk is sodium potassium ATPase pump, because you know the most of the energy of our body is utilized or spent in those sodium potassium ATPase. Because it is very important to throw sodium out and out and out. Okay, but when the sodium, when the oxygen is too less, like severe hypoxia. Okay, the sodium potassium is already destroyed, that system is already destroyed. Now the system which work next to it with a very less energy is the calcium channel. So in the irreversible cell injury, the calcium channel is not working. The calcium channel is even not working. The calcium channel work with a very less energy, but still the calcium channel is not working because of this severe injury, because of this irreversible cell injury. Now what is calcium? Calcium, it can activate lots of enzyme inside the cell, including mitochondrial, including apoptotic enzyme, including uh, enzyme activation like lysosomal enzyme, phospholipase enzyme, nuclease enzyme. So the calcium can activate lot of enzymes which are not good for our cell. Okay, also one thing we need to know that the calcium which leaked from outside to inside of the cell, this is not the main common cause of causing injury. The common cause is when the calcium comes and the calcium is not thrown out of the cell, there is a buildup of the calcium, and this calcium is stimulating the endoplasmic reticulum, and the endoplasmic reticulum release the calcium which is present inside it. And this is the calcium which the calcium which is released by endoplasmic reticulum. This calcium is responsible to increase the calcium level inside the cell tremendously, inside the cell tremendously. What calcium does, the cytochrome c which is present in mitochondria will leak out and which will cause apoptosis, okay, which will cause apoptosis. And apoptosis is cause cell death. Apoptosis is causing cell death. On the other hand, what is happening, due to less ATP, actually this is related to this, all things are related, just different differences between each. Okay, mitochondria, because of the calcium, cytochrome C is released or can come out in the cytoplasm and causing apoptosis, or causing activation of an apoptotic activating enzymes and leading to cell death. But the mitochondria not functioning properly, less oxygen is supplying and less ATP is formed, which will cause decrease in the ATP will lead to hypoxia, and the energy dependent calcium channel is not working, and the calcium buildup will take place. In this calcium will lead to the activation of enzyme like lysosomal enzyme, like phospho phospholipase, like nuclease. Okay, so these lysosomal enzyme, we know lysosomal enzyme, it is enzyme from lysosome, and lysosome is known as suicidal bag. Okay, suicidal bag. This is most important enzyme to me remember in necrosis. Okay, in necrosis, most important enzyme in necrosis is the lysosomal enzyme, and causing cell death. How it cause death, I hope you guys know. The lysosomal enzyme is secreted, which is a hydrolytic enzyme, and the vacuoles, it is secreted in a form of a vacuole. And what it does, the vacuoles hug, or vacuoles collide with the cellular component, and gradually it degrades, it kills the whole cell. Okay, kills the whole cell. Another enzyme which we talk about is a, let's talk about the nucleus enzyme. In nucleus enzyme, it is causing the degeneration of the nucleus. We saw one thing in cellular, in reversible injury, that the cell is, the nucleus is, we can find the coagulate, like con- condensed genetic material or a condensed chromatin material in the reversible, but it is not the case in irreversible. Suppose if this is the normal nucleus, the nucleus in irreversible, where the cell want to die itself, will be killed in three ways, either of the three way. First of all, pycnosis means it is condensed in single one dot. The whole genetic material is condensed in one dot, which is known as pycnosis. Okay, which is known as pycnosis. Because of this appearance, it is also known as ink drop appearance. Okay, as it a drop of ink fell, this is known as ink drop appearance, pycnosis. Another is a karyoraxis. Karyoraxis. In these, the genetic material is kind of a fragmented in small fragment, or maybe large fragment, and this is how the nucleus destroyed the nucleus. This is karyoraxis. The last one, and this is the most dangerous one, is the karyolysis. It means the complete degeneration, complete destroy of the nucleus, and it is just converted into small, very small, all the nucleus or genetic material is destroyed in small, very small particles. This is called a karyolysis. Okay, it is lysed into small pieces. How to differentiate between the reversible or irreversible, or apoptosis or normal nucleus, how it is appeared on the base of nucleus. So one procedure is done, which is known as gel electrophoresis. Okay, gel electrophoresis. In that, in normal nucleus it will be appear as a single band. So in normal nucleus, if you do the gel electrophoresis, it will appear just a single band. Okay, just a single band. But in case of a necrotic nucleus, it will appear as a smear pattern, like we smeared on a anything, on a culture plate, as we do smearing. The smear pattern will occur. But in apoptosis, the genetic material is fragmented into small pieces. Like this, if this is the genetic material, it is cut it into small pieces. So when the gel electrophoresis is done on apoptosis, it will appear like this. Okay, so it appears as a ladder. So this is also known as tram track appearance, or step ladder appearance, okay. So these are finding in nucleus is doing this thing. It is destroying the nucleus. Lysosomal enzyme, lysosome causing the cell death by hydrolytic enzyme and all. The last thing which remember is a phospholipase. I need to talk a little bit more about this, so we will do this in the next. Okay.